In the previous sections, we learned the primary structure of DNA. In this section, we will learn what is the DNA double helix and how the DNAs are replicated. So the learning goal of this section is to learn how to describe the double helix of DNA and how to describe the process of the DNA replication. Now, we know that the DNAs contains adenine and guanine, which are purine bases, and cytosine and thymine, which are pyrimidine bases. Okay. Um, Now, it has been experimentally determined that um, always this adenine base is paired with thymine base and guanine base is paired with cytosine base in the DNA molecules. How did they understood that? So they have been noticed that there is a 1-to-1 ratio of adenine and thymine and 1-to-1 ratio of guanine and cytosine. 1-to-1 ratio means these pairs, the bases that are in these pairs exist in the same amount. Now, we can summarize this relationship as since the adenine is the ratio in between adenine and thymine are 1-to-1 and guanine and cytosine are 1-to-1, we can say that there are same number of adenine bases and same number of thymine bases and same number of guanine as same number of cytosine. In other words, we can say there are same number of purine molecules to same number of pyrimidine molecules. So this relationship has been determined experimentally when analyzing DNA molecules. And then finally, we can say that a number of adenines are equals to a number of thymines and number of guanines are equal to number of cytosine. So how would this be possible? So this can be possible when there is a complementary base pairing occurred in between two single strands of DNAs. Now, what is this complementary base pairs means? So, as I mentioned, when adenine is always pairing with thymine, we call this adenine and thymine as a complementary base pair. And this is the adenine base and this is the thymine base in the DNA molecules. Now, we know that the base is attached to this sugar in DNA chain, right? And this comes from one nucleotide. And this comes from another nucleotide. And when there are two nucleotides that contains the adenine base and the thymine base, they can make two hydrogen bonds in between the hydrogen atoms and the other electronegative atoms. So when making these hydrogen bonds, they will interact together and they will just bind together. And this is called base pairing. So the base pairing occurs as a result of hydrogen bonds. Okay. Now, in particularly, adenine and thymine will make two hydrogen bonds based on their structures. Okay, that's something you need to memorize. Adenine always pairs with thymine by two hydrogen bonds. And there's another complementary base pair, guanine and cytosine. The guanine and cytosine will make three hydrogen bonds. Okay, so this base pairing is a little bit stronger than adenine and thymine because they have three bonds. And this is the other complementary base pairing. And these are linked by, again, three hydrogen bonds. So what we need to learn is that adenine adenine base will interact with thymine, but these are very specific base pairing or very specific complementary base pairing. Adenine will not make pairing with cytosine. Okay, this will not happen. And adenine will not make a base pairing with guanine. That is not possible. Adenine will only make a base pairing with thymine. Just like that, guanine will only make a base pairing with cytosine. So this is why we call them as complementary base pairs. Now we'll see the structure of the double helix of DNA. This double helix occurs due to this complementary base pairing. Okay. Now the double helix consists of two primary DNA strands. Okay. These two primary DNA strands have just held together by the hydrogen bonds in between the complementary base pairs, which are adenine, thymine, and guanine and cytosine. And we know that this sugar phosphate backbone is showing here in this purple color ribbon, and you can see the complementary bases in between the two strands. Now, the double helix of DNA is just um, like a spiral. It's like a spiral staircase. If you're just thinking this about in as a three-dimensional structure, it looks like a spiral staircase where the sugar phosphate backbone would just be considered as the railing of the stairs. Okay, and these complementary base pairs will just can be considered as the steps of the stairs. That is the double helix of DNA. Okay, now let's do a learning check, and this is one learning goal of this class. Write the complementary base sequence for the matching strand in the DNA section shown below. Okay, there is one thing that I need to mention. Um, so this shows a single strand of DNA. Okay, this shows a single strand of DNA, and which goes from the 5' end to 3' end. So when one end of the double helix, when the one end of a single strand is 5' and the other end of the opposite strand will be 3'. Okay, so the 5' to 3' direction of one strand will go like this, and the other strand will have the opposite direction where it goes. The 5' to 3' will just go like this. Okay, so if one end of the so this is one of the end of this double helix, right? So the two strands will have opposite ends. One will be 5' and the other one will be 3'. Okay, so when you are writing the complementary base sequences, now you need to know that if one single strand has the base sequences like this, where this end has 5' of this sequence, then we need to know the other sequence should have 3' at the same side. Okay, and this one will have 5' um, on the other side. So that's something you should remember when you're writing the complementary base sequence for the matching strand. So the complementary strand can be written by just by keeping this as a note. Adenine always pairs with thymine and guanine always pairs with cytosine. Okay, this is very simple. Just remember this as ATGC. Okay, ATGC. Now we have adenine here, then you just have to write down the complementary base for adenine, and for guanine it's cytosine, and thymine will bind with adenine and cytosine will bind with guanine. Just like that, keep writing the sequence, okay, and then you will get the answer like this. So these are two complementary strands with the correct complementary base sequence. And this is how the bases are bonded to each other using hydrogen bonds. Now we'll see what is this DNA replication and why it is needed. And we know that this DNA contains very important genetic information as a base sequence. Okay, and this base sequence just um, store all the genetic information. Now, the function of DNA is to preserve this genetic information and to transfer that genetic information to the new cells. So, in any living organisms, we know that there are millions of cells in our body, and these cells are divide at each second, right? They will divide to make more cells or new cells. Now, the it's very important for this genetic information to transfer to the new cells. And how these are transferred? They will be just transferred as a new copy of the DNA molecule. So, for example, if there are a certain number of DNA molecules in a cell, so those will be copied and will send the same set of DNA molecules to the new cells. So this is called DNA replication. So the two newly synthesized DNA strands, which are called daughter DNA strands 1 and 2. So these two are the same exact copies of this parent DNA molecule. So how does that happen? That happens due to the complementary base pairing. And after this, the parent DNA is unwound or separated, the new strands will be generated by complementary base pairing to make sure that the two new strands will be the same exact copies as parent DNA molecules. Okay, so this is what happens in DNA replication. So the strands in the original or the parent DNA molecules will separate. Number one. So as they are separated, just that happens by unwinding the double helix. So unwinding occurs by breaking up those hydrogen bonding in between the complementary bases. Now, after it unwinds, um, the two strands that comes from the parent DNAs will be separated, and the two strands will act as the templates for the synthesis of new complementary strands of DNAs. So the purple color of these two strands are showing the template, the DNA templates to make the new strands. After the parent DNA molecule is separated to expose the DNA templates to form the new strands, at this stage, the nucleoside triphosphates that contains in the nucleus, which are deoxyadenosine triphosphate, deoxythymidine triphosphate, and deoxyguanosine triphosphate, and deoxycytidine triphosphates. So we discussed about these nucleotides in the previous section. So these molecules will just come and pair with the bases according to the complementary base pairing rules that I mentioned before, which means the cytidine, the cytosine will come binds with the guanine, and the thymine will pair with adenine. Just like that, these small nucleotides just comes and attach with the complementary bases. Now, at the same time, in order to make a new strand, another bond has to be occurred to make the phosphodiester backbone, right? So the phosphodiester linkages will formed at the same time. So there should be two bonds need to happen. Hydrogen bonding in between the complementary bases and the phosphodiester linkages to connect the nucleotides with each other. So that's how it's going to start making the new strand. Now we will see a little bit details about this DNA replication process. Now the first step of this DNA replication process is the unwind of the DNA parent strand. So this unwinding occurs by an enzyme called a helicase enzyme. So this is an important term, helicase enzyme. Okay. The helicase enzyme is showing in here in this orange color. And once this helicase enzyme binds to the parent DNA molecule, um, it can just break up those hydrogen bonding in between complementary bases to separate the two strands. And this process can happen at multiple places of the same DNA molecule. Okay, not just one side. It can happen at multiple sections. Now, once this helicase unwinds unwinds this double helical strands to separate the two strands, the second enzyme comes into place called DNA polymerase. Now, this DNA polymerase catalyzes the replication process, and the place where that DNA polymerase catalyzes this process is called as the replication fork. For example, in this section, so this is one of those sections that you are seeing here. So just consider that as one of the sections from the parent molecule. And so in this section, the DNA polymerase catalyzes the replication right here. So we call this site as the replication fork. Now this DNA polymerase enzyme has a very picky moving direction. So it only moves in the 5' to 3' direction of the newly synthesized DNA strand. Okay. Now you see this purple color strand as the parent strand and this blue strand as the newly synthesized strand, one of the newly synthesized strand. So in the newly synthesized strand, you see the 5' end is in this side. So the 3' end should be in this side, right? So this DNA polymerase will only move to this direction of the newly synthesized strand. So the role of the DNA polymerase is to form that new phosphodiester linkages in between the nucleotides. Okay, the nucleotides will come and attach to these complementary bases, and DNA polymerase will make those phosphodiester linkages to make it a actual DNA strand. Now, the new DNA strand will grow from 5' to 3' direction, right? So this new strand here is called as the leading strand. Remember, so this strand will just grow from 5' to 3' direction, and that strand is called as the leading strand. So the leading strand can grow continuously from 5' to 3' direction in the same direction that DNA polymerase can move. The other new DNA strand, which is the lagging strand, is synthesized discontinuously in short, separate segments. To initiate on the lagging strand, first an enzyme called primase enzyme will produce short RNA fragments, and these RNA fragments are called primers. The RNA primers will serve as the starting position for DNA polymerase. And the DNA polymerase will begin extending the complementary DNA strand or complementary DNA fragments at the 3' end of the the new strand until the 5' end. Um, as a result, the DNA replication on the lagging strand consists of DNA segments called Okazaki fragments. So I mentioned that this is not a continuous process, right? So this discontinuation occurs due to the direction of this DNA polymerase moves. So it moves from 5' to 3' direction. But the new strand need to be formed from 3' to 5' direction. So these Okazaki fragments are resulted due to these opposite directions. Um, and the DNA polymerase will eventually replaces the RNA primers in each segment with the corresponding DNA segments. And finally, there's another enzyme called DNA ligase. DNA ligase will just connect all the Okazaki fragments, the sections of newly synthesized DNA fragments together by the phosphodiester linkages. And this newly synthesized strand is called the daughter lagging strand. So the enzymes and proteins are required for this process. And we will learn what are those enzymes and proteins involved in the DNA replication process, and it's summarized here. First, the helicase enzyme will break the hydrogen bonds in between the complementary base sequences, the parent DNA strand at the replication forks. So this will give two separate DNA strands. Then a protein called single strand binding protein will attach to the separated parent strands to keep them apart, and the bases will exposed. Now there are two types of strands that we discussed. The leading strand that moves from 5' to 3' direction, and the lagging strand moves from 3' to 5' direction. The primase enzyme only involves to the lagging strand. Okay, the primase will synthesize those short RNA segments called primers, which are starting points for DNA polymerase enzyme. That is the role of the primase. DNA polymerase polymerizes both leading strand and lagging strand. So it will catalyzes the formation of the phosphodiester linkages on the new DNA strands. So that is the role of the DNA polymerase enzyme. It catalyzes, this is an enzyme, it catalyzes the formation of phosphodiester linkages on the new DNA strands between the 3' ends of the growing chains and available complementary nucleotides. And also DNA polymerase adds nucleotides continuously in the 5' to 3' direction to a growing DNA chain. That's what it does for leading strand. It makes this continuous leading strand from 5' to 3' direction. In the lagging strand, at each primer, DNA polymerase forms those Okazaki fragments until it reaches the next primer and stops. The last enzyme was DNA ligase, and this only works for the lagging strand. It doesn't involve for leading strand because no Okazaki fragments in the leading strand. In the lagging strand, it joins all the Okazaki fragments. So these two daughter strands work differently due to their different directions, and therefore it involves multiple different types of enzymes as well. Okay, now let's do a quick learning check. Match the following to their definitions. A short segments formed on the lagging strand. What are those short segments that are formed on the lagging strand? Just try to find the answer from what you understood. The second one is the starting point for synthesis in unwound DNA section. What is the starting point from these four keywords? The enzyme that unwinds the DNA double helix. The enzyme, it has to be helicase or DNA polymerase. So what is that? The last one is the enzyme that catalyzes the formation of phosphodiester bonds of complementary bases. So what is that? So this is a very important question. So you should be able to um, identify what are the keywords and what are the enzymes involved and their roles and what are the definitions for these different keywords. So let's see the answers. The answers are given here. You will have similar questions in your homework as well. So this is the end of this section, and there's an animation video which describes the DNA replication process. So it will help for you to understand this process further. Thank you.